Right, you're very welcome to this first episode of this interview style video where I'm going to chat to different researchers about their work, what they're doing outside of their research, and, well, maybe their love of the superhero genre, if they have it. And my first guest for this first episode is Alex Cloherty, who is a PhD candidate at the Amsterdam UMC in the Netherlands. Alex, you're very welcome.
Thanks so much for the chat today, Barry. I'm looking forward to it.
Excellent. It's great. It's great that you've accepted my request or my invitation to be part of this. This is the first one, so it could be, a little bit of experimentation, which is what your research is all about. Can you just briefly maybe give people a little bit more information about you and what you do before we get into some of the questions?
Yeah, definitely. So, as you already said, I'm deep into my PhD at the Amsterdam UMC, and I'm in an immunology department, but I also work a lot with viruses. So I'm kind of walking between these two different fields of immunology and virology. And my project focuses especially on how autophagy, the decluttering system of our cells, can be sort of harnessed in order to protect us from the different viruses I study, which at the moment are HIV dengue virus and now also SARS-CoV-2.
Right, well, we'll get into a little bit about that later on, and also the whole process of decluttering. This idea of I think of it like a rubbish truck that just collects junk and gets rid of it, or maybe recycles some of it, of course, because it's not all disposable, right? It's not all just thrown away.
Exactly.
We'll get into that a little bit. So let's jump straight into then a little bit on your research then. Well, you've mentioned it there. We find ourselves in these unprecedented times. And for someone who's working on microbes and viruses, as you've just mentioned, this must be very exciting, but also concerning at the same time. But before we talk about your research, why are you studying microbes? Were you always interested in microbes?
I was always interested in science, and I had some great high school and elementary school and university professors who really, deepened my love for biology especially. Microbes, I never really thought about them much until I got into university. I mean, I always liked bread, but I didn't really know that microbes had such a role there. I think I really started getting interested in microbes when I took a mandatory course, Introduction to Microbiology, at University of British Columbia back in Canada, where I did my bachelor. And I had this awesome professor who not only taught the course, but she would also have regular segments called Microbes in the News. So she would talk there about points at which the microbes were coming up in news across Canada and across the rest of the world. So that really hooked me that microbes had such an impact on the human experience, because I was also always interested in history, and she kind of brought together history, current events, and microbiology, and I thought, this is so cool, I have to know more about this.
Oh, it's very interesting that, in fact, you were drawn into the area of microbes thanks to some very innovative science communication, in fact.
Exactly.
That's what your previous lecturer was doing at the University of British Columbia. So I think that's quite nice, given that's where you find yourself and some of the things you're also involved with. But when you were at school, I have to know this because you're in biology, were you the kid who loved to do all the dissection experiments?
I was that kid. Yeah, I was the nerd of the class always.
What's the one thing when you were doing dissection experiments? What's the one thing that you always remember dissecting and thinking that was the coolest thing that I have dissected up until now?
Actually, I think the coolest thing was dissecting owl feces at elementary school. So, sounds really weird, but owls swallow, if I remember this correctly, this is like at least 20 years ago now. If I remember this correctly, owls swallow a lot of their prey more or less whole. So digging through this
just
owl
like a snake.
Yeah.
Just like a snake, basically. Yeah, okay.
Yeah, exactly.
Right.
But I was coming across these like almost complete mouse skeletons and all these, quite large chunks of animals, and I thought, this is amazing. Like, you can go all the way back through the owls' meals over the last week or so and get quite a complete picture of what the animals were. So it was kind of like, dissecting owl poop, but also dissecting mice and other rodents along the way. Yeah.
I got just a logistical question. I mean, question I think people would like to know the answer to. Where do you get owl feces from?
That is an excellent question that I have no idea as to the answer.
Is someone collecting owl feces for schools? Is that what they were doing?
I guess so. So I was at this great school in Israel at the time. My dad was a diplomat, so we moved to Israel for a while when I was a kid. It was an American international school, and there are very few kids per classroom. So I think it's probably not a normal thing. They must have had some kind of connection with a museum or, I don't know, an animal sanctuary or something, because yeah, it's quite special to get to dissect owl poop at like age 7 or whatever I was at that point. It was a very special experience, though. It stays with me to this day.
well, obviously it has made a serious impact, of course, seemingly, given your career choice. At 7 years old, was I dissecting any animal feces? I'm going to say no, I wasn't. Even though my father raises pigeons, and we have pigeon lofts down the back of the house, and I would, when I was younger, see lots of pigeons, and of course when you have lots of pigeons, you have lots of feces. So I would have seen a lot of that, but never dissected it. So that's interesting.
Not so many
very,
skeletons
very
in pigeon feces, though. I guess
No,
I would imagine
no.
it's less interesting.
No, pigeons are not known for swallowing mice or rats whole. That's not their diets. Let's just say that. Now, I'm not a biologist, I'm a physicist, but I've kind of delved into the area of biology in recent years and done a lot of research on it. I think one of the facts that always jumps out at me is in relation to bacterial cells and in comparison to how many cells there are in the human body. And I think that the number is, anyway, according to current research, is that there are at least as many bacterial cells in the human body as there are cells that make you up. So the cells you find in your brain or in your liver or in your blood. And then the thing is, when people, they hear the word bacteria, though, they immediately think about bad. But there's lots of good bacteria inside our bodies. We've got the bacteria in the gut that help us to digest food. There's even bacteria that occupy little pores in the skin to prevent infection or anything from getting into the body. Why do you think then that people focus on the bad bacteria more than the good? Is it just a case that the bad bacteria have got better PR?
Yeah, I think it comes down to news coverage in the end. I mean, if you look at any news stories across the world, you do often see the bad more than the good, right? So that's part of why I wanted to start Microbial Mondays, is to be kind of a good news movement against all of the bad news coming out about bad bacteria. I mean, it's kind of like car crash television, right? If you see all these stories about bad bacteria in your lettuce, and there's a plague, anthrax letters, it's all bad bacteria. You can't take your eyes away from it. But maybe it grabs your attention a little bit less to know that, bacteria are important for your bread or your beer. But I think it's just as important and just as interesting once you dive into it.
Yeah, I think, do you know what? I think would people drink as much beer if they knew that bacteria was behind its production? I wonder.
Good question. I mean, it's mostly yeast for beer, to be fair, which maybe people have a little bit less of a bias against. But yeah, I mean, would you stop drinking beer if you knew about the microbes behind it? I mean, the taste is pretty good. I hope the answer is no to that.
Yeah, I mean, in general, the taste is pretty good. There are some awful beers out there, of course. Let's put that out there. Not naming any certain beers in the Netherlands, so there might be some people might take offence to said beers. And just thinking about that, I think that's a really good point in terms of the news element, that you'll never hear opening headline on evening news or a headline in a newspaper when you read it the next morning will be, certain bacteria helps your gut to digest food in ways that will prolong your life. It's always, as you say, the ones that may be associated with a condition that could have ill effects for somebody. How can this be reversed? How can we think about making sure people know more about the good, and not to say that they shouldn't know about the bad either, but how do we make sure people know more about the good that bacteria do?
That's a tough question. I mean, in the end, it kind of comes down to clicks, what news agencies will report, right? And from what I understand, bad news or bad reviews tend to get more clicks. So maybe it's kind of a change in mindset that's needed, but that's hard for a couple of lone scientists to do. But I guess just good science communication in the end, because it's easy to make bad news interesting and connect it to yourself. But I think it can take a little bit more creativity to make good news or interesting findings interesting to somebody who's not studied it themselves, who's not spent 20 odd years of their lives diving into this one subject. So yeah, I think good science communication is the way to do it. Keep it interesting, keep it fun, and then people want to read about it. There's this Instagram page that I love called the Good News Movement, and they do basically this, but with news in general. So that's kind of an inspiration for me in trying to get good bacterial news out there as well.
That's a nice resource. Good that you mentioned it as well. That's a very nice bridge on to the next section then, which is the idea of science communication. We're speaking right now because we know each other through science communication. And I don't know about you, but when I was an undergraduate, I wasn't thinking too proactively about science communication. It wasn't something that was always on my mind. I have a physics background. I was studying a bachelor's degree in physics. I was doing experiments. I was working on equations, and I was looking at exciting physics concepts. That was what I really was thinking about. But then I started to think a little bit more about science communication, particularly when I started my own PhD, which is, how long ago is that? If I give my age here, people will know. I'd say it's about 17 years ago. There we go. From your perspective, when did you start engaging with science communication?
Science communication, to some extent, was actually a big part of my academic experience the whole way through. So yeah, maybe somewhere between science communication and education. So even as a high school student, I used to tutor younger students in math and biology and chemistry, and I really loved it. I loved teaching. And when I got to university at UBC, there was this cool program called supplemental learning, in which higher level students would basically break down the material in a more understandable way, and then kind of at a bit of a slower pace than in the lectures for introductory students. And I got recruited to be one of the supplemental learning leaders, to be one of the people breaking down that material. And it was just the best. I loved it so much. It wasn't in a lecturing format as well, so we really tried to make the material fun, and we would design games around like how molecules interacted in organic chemistry, or like quiz show style formats for microbiology, for people to help remember the names of all of the different microbes that they needed to know for the finals. And actually, I really missed that when I moved to the Netherlands for my master, and that's part of the reason I started my blog, because I thought I want to have this sort of science communication, education component in my life still. So it's been a big part of my experience, actually.
That's actually refreshing, because of course I'm from another generation when it wasn't something that was, let's say, focused upon. It wasn't something that was integrated into the education track or the communication track. There wasn't necessarily anything there that I could say, yes, this was science communication related. So it's actually good for that, to hear that. And for those also who are wondering, you've mentioned Microbial Mondays on your blog. Let's tie those together. Your blog is Microbial Mondays, because you mentioned Microbial Mondays earlier on. In terms of maths or science communication, right, my earliest memory of maths communication, in particular maths communication role models, has to be the Count from Sesame Street, who would be helping people to count whatever they were counting, apples, oranges, whatever. I thought he was great, right? But I mean, it was no fun if he didn't go beyond 5, because at least you get to hear him say a number of times. And if he's just doing a 3, you're like, you're kind of getting so short if he's not doing it like, at least 6 or 7 times. My earliest memory, though, of science communication, in terms of having somebody do it in person with me or with me and my peers, is probably from my teenage years when we visited a science museum during a school trip. But the times have certainly changed in terms of science communication, as you just mentioned. And I think maybe this might go back to the previous answer, but maybe just to get a little bit more detail, what's your earliest memory of meeting or speaking to a science communicator?
Ooh, a science communicator. The first thing that jumps out in my mind is a teacher who was just a great biology teacher. I don't know if that counts, but he had us bake. He had us bake cakes and then design the cake to represent the interior of a cell, and that got me really hooked on cellular biology. So I think that was my first introduction to really effective science communication that wasn't just out of a textbook.
And where was that? Was that in your teenage years, or was it when you were still in primary school?
It was in high school, yeah. I think I was around 12 or so. And he, every lesson, he had one of these, little experiments or projects. We also made, Coke and Mentos rockets and, all of the classic ones he had us do.
Yeah. I did that. We did that as well during the PhD. At the start of the PhD, we used to do the Mentos Coke thing inside in the office. Maybe I shouldn't be saying. Maybe I shouldn't be saying that. Inside the office. Cleaning it up. Yeah. Well, fortunately, that office was taken off us during the PhD, and it was given to architectures at the University of Limerick. So the architecture department. So we kind of didn't have to worry about any potential messes there were. It does. We can confirm it does do what it's supposed to do. Left a couple of marks on the ceiling, but done that too.
Yeah, it's quite dramatic, yeah.
It is quite dramatic, yeah, that's a good way of putting it. And then just thinking a little bit more about the science communication element. I've been an active science communicator for many years, even though I haven't, let's say, had structured introduction to science communication in my formative years as it seems you would have had. And in the past, people I would have told about what I was doing would have had different reactions to what I was doing. So some people would have said, that's great, so cool you're doing this. Some people just said, look, I don't know what that is. Can you just explain it to me and what exactly you're doing? But then some people just, and I have to say this is still the case, don't see the point of science communication, and they don't see the value of science communication. In your career, whether it's your research career or in the work you do in science communication, have you met people who fall into all of those three categories?
I would say the first two more than the third, actually. I think maybe it's partially because of the research that I do, especially because it's so related to health. People, I think it's easy for people to make the connection as to why that's important. If there is a new, if a new scary virus is coming out, people want to know about that. It's quite a clear connection to themselves. But I definitely have had to explain to people, the different areas of science communication as well. Some people think, does that mean you're selling companies' products to people? Like you're going to be advertising drugs and how they work? Or that's usually the first understanding, I think, that people have of science communication. And yeah, oftentimes people don't realize that there are so many people behind the scenes making research understandable, whether it's by, the Nature News articles or places like your YouTube channel or, it's a bit behind the scenes.
Absolutely, yeah. I'm just from my own perspective, I did meet a lot of people at the start of the process who were like, what's the point of this? And I mean, it's starting to change. And what I think about the people who were saying, what's the point of it, was mainly those who I would have encountered or crossed paths with in the academic world, because I was a postdoc and I was doing the science communication in parallel to my work as a postdoctoral researcher. And every now and then I would meet somebody who would kind of say to me, Barry, your research is very good, but why are you also spending time on this science communication element? Why do you think that's a worthwhile venture for you to embark on? And I guess the reason I have to ask you that question is that I would expect that nowadays science communication is seen as more inherent to the science process, to the research process. And if somebody says they're also doing some science communication or have embarked on a small project in relation to it, it's more accepted now that it's part of the process. So for example, maybe just asking it more generally, for you then at the Amsterdam UMC, do you also see that with your colleagues? So some of your colleagues who are there, are they also branching into the areas of science communication and more frequently than perhaps their predecessors would have done?
Yeah, that's really interesting to hear your perspective because I haven't had that reaction even from the older PIs or the most experienced PIs in my department have been pretty supportive as well and thought, the whole FameLab thing was really cool. So I guess the approach has changed. And yeah, definitely other people around the Amsterdam UMC are also getting more and more into science communication. Like a colleague from my master, who's also now doing a PhD there, was recently on the news explaining Omicron. He's a virologist as well, so on the Dutch news. So very cool, yeah. And others at the Amsterdam UMC have started a podcast called the Struggling Scientist Podcast. So there's quite some science communication going on. I think more amongst PhD students or postdocs versus PIs because I guess they have other things to do.
Of course they do, yeah.
Yeah. But it's definitely alive and well, science communication at the Amsterdam UMC.
That's very good. Well, actually, I think you made a good point earlier on. I think it could also be the research area, and it depends on the research area you're working on. And perhaps given the current situation we find ourselves in, with everything that's been going on over the last 2 years, that the need for that is very much there. We'll talk about that again in a few minutes, but I think the need is very much there for that. And you have created very nicely for me an effective bridge to section number 3, and that's to talk a little bit more about your experience recently in regards to science communication in the FameLab competition, and also we can chat a little bit more about your research. So for those who don't know, Alex here is the winner of the International FameLab competition for 2021. So that's absolutely brilliant. Fantastic. You are crowned the winner across the world, basically. And I don't know how many countries took part, 20 or 30
23.
Countries had 23. There we go. 23. Now, for those who don't know what FameLab is, here's the brief synopsis. FameLab is a science communication competition where contestants are given 3 minutes to pitch a scientific idea, concept, or research area, and they're not allowed to use slides. However, the contestants can use props to support their story and to use them in informing this engaging or interesting narrative in those 3 minutes about something that could be very, very complicated. Normally, over the years, FameLab would take place in person and the international final would be in Cheltenham. This year, unfortunately, due to the whole situation we have with the pandemic, it took place online. Now, Alex, you pitched 4 videos during your FameLab 2021 journey. You had one at Amsterdam UMC, you had one in the Dutch national final, which was hosted by the Eindhoven University of Technology, and you had 2 videos in the international FameLab final. And that's the one thing I want to start off with, looking at how things went and looking at the videos. How different do you think the experience of FameLab would have been if you had presented the pitches to live in-person audiences during the FameLab 2021 competition?
I imagine quite different. Actually, the first pitch at the Amsterdam UMC was live, so I did have a very little bit of that experience. So they rented out a massive lecture theater, and we were all sitting like 4 meters apart from each other during the early summer of this year when it was possible.
But how many people were in attendance for that?
There was almost no audience. I think there were maybe 4 people who were allowed in. So it was still different than it would have been without coronavirus, for sure. But yeah, already that was quite a different experience. I am a total Luddite at heart, so I've really been trying to embrace technology over the course of this. So for me, actually, the first one was easiest to stand up on a stage. I grew up playing a lot of music and performing a lot musically. So I'm quite comfortable on a stage, actually. So
okay, so what instruments do you play?
I play bass guitar, ukulele, and a little bit of guitar. And I used to play piano, but I don't have a piano here, so I'm extremely out of practice on that one.
That's cool. You've still got a bass guitar and a ukulele and a guitar?
Yeah, those I have here. Yeah.
Wow, that's great. Yeah, excellent. So obviously, I mean, I've also been in a band and played on stage, and I can tell you that it's a huge benefit, and it helps you with your ability to deal with being on stage. So I can imagine then that musical background and experience would have been a huge asset to you when you were doing the first pitch first pitch in person at Amsterdam UMC. So when it turned out we were moving to the online situation for the national final, were you disappointed?
Not really. I had prepared myself for that. I thought, okay, probably that's, I mean, as more and more people came in, it was clear that, it was clear that things were going to have to go online. So it was what I expected. I think for me, it was actually a really good experience to get used to using more technology in my approach to science communication. It sort of forced me to meet my fears in a way that is really useful in this day and age. I mean, some of the best science communication I've seen is on YouTube or otherwise online. So it was a good way to force me to get into using video for science communication.
Yeah, well, I mean, I had the same issue as well in 2020. I had a whole bunch of stuff cancelled when the first lockdown struck in March 2020. And yeah, a lot of my events were cancelled, and then suddenly I got contacted to do an event for a book festival, and they said, will you do it online? I just said, absolutely. I didn't know how I was going to do it, but I just agreed to it. And then once you're backed yourself into a corner, you have to reverse engineer how you're going to deliver it. And that's how I ended up embracing the online environment. So kind of forced myself, and as you mentioned yourself as well, that you're kind of forced to think proactively about how do I come up with a solution that would work for myself, but also for my potential audiences. Now, in your videos, 3 videos and your live pitch, you would have presented about your own work across those 4 pitches. I have to ask this question though, if you could have pitched on a topic outside of your field in FameLab 2021, what would it have been?
Ooh, really good question. Like outside of my field, as in not my research or not microbiology?
Yeah, let's get real specific. I'll give you two possible answers, right? Let's give you the easy one first. Outside of microbiology.
Okay, I would say that's the harder one for me because I really like microbiology. Probably organic chemistry. I really like organic chemistry as well, and I know it scares a lot of people. Definitely in undergraduate degrees, at least at UBC, it was like the scariest course that was mandatory for biologists to take. But I always really liked it.
I think it probably got scary because there's the word mandatory in it. So when people hear that you have to do it, you're like, oh no, this means it's really hard.
Yeah, that might have had something to do with it. That's true. There's also an air of mathematics about it, I think. There's, like, they talk about equations, and that can be quite scary for biologists. Yes. So, yeah, the combination of the two was terrifying.
Okay, so, and then any other topics that you would, is there anything that you would do if you were, given a clean slate and you thought, oh, well, I'd love to talk about this? Is there anything what would you feel comfortable talking about outside of the biological area or even organic chemistry?
Outside of biology, let's see. That's a really good question. I think I probably would have chosen something that has some interesting historical background and come at it from a history point of view. I don't know, like some story about Isaac Newton and how he had his aha moment with the apple falling from a tree. I think I would have come at it like that because I really love reading about the history of science.
And spoiler alert on that, the apple did not hit him in the head. There was no such apple that hit him on the head.
Okay, so I have to do more reading on that before I present at FameLab.
Yeah, I've done the research. I did the research for one of my books, and I dug it up and found the evidence that clearly states that the apple did not hit him on the head. So that's just a fable in science, a myth. Reversing it for me, I mean, it's probably easy for me because I do a lot of research and read lots of papers in different areas. I would probably have done, if I was doing it, even though I'm a physicist, I would have done it on CRISPR. That's what I would have done. Because, I mean, that's something I find fascinating, the idea of being able to rearrange DNA and move things around and, take things in, take things out, put something else in. Or I would have done it on ethics, science. Maybe that's something I would have looked at as well, which is also kind of related to your area, but you could, if you push it to the boundaries, you step further outside of your field because you also have to deal with ethics in terms of your experiments, of course, don't you?
Yeah, definitely. Yeah, we use human tissues in our experiments, so all of the paperwork for that has to be carefully in line. Yeah.
Yeah, so you could have done it on ethics as well, you see.
Yeah, true.
Yeah. we've seen a lot of science communication over the last 2 years about viruses, microbes in the media. It's just the world we live in at the moment. How much of it makes you cringe when you see it, read it, or listen to it?
I quite carefully filter who I listen to science communication about in terms of microbiology. So in terms of the science communication that I allow myself to be exposed to, it's pretty high level. But yeah, if I delve outside of that, there's quite some cringing going on. I've had a lot of evenings where I read something in the news and I have a little hissy fit at home that my poor partner has to endure when I'm complaining about how other people are writing about stuff they don't understand. And yeah, it's ups and downs.
Yes, of course. I can imagine that when you see something that's in the news, it can be quite infuriating, given that, I see it as well, that there's a lot of things that are factually questionable and also things that are very much conjecture. Then when you start to reflect about how you can address this, do you use your blog, Microbial Mondays, to try and address this and to maybe put the scientific fact out there associated with a story, and then directly relate to that, link or connect to that story? Or do you do it more, let's say, less obviously, calling it something into question and maybe write a blog entry that just says, okay, here's a certain topic in microbiology. This is the 101, you got to know about it.
Yeah, more the latter, because I do understand the difficulty of it, especially if you haven't got a PhD in microbiology. I mean, it is tough to sort through the information and the misinformation that's online. I mean, like this weekend, I'm looking through some soil microbiology articles, and it's not exactly my field, but it's still microbiology, and there's a lot of pseudoscience out there as well, which it can be quite difficult to separate from solid science. So I don't want to shame anybody for accidentally bringing forth the incorrect information, but I do try to, in my way, correct it in blog articles without sort of throwing any shade at any particular person.
Yes, of course. The other side of the coin is that there are certain instances where if something is blatantly wrong, then it needs to be explicitly highlighted for that fact. And I've seen many things where I've just called into question. I do it in some of my presentations. I see things and I bring it into the talks and I just say, look at this. This is nonsense. This is complete and utter. Do not listen to this. This is wrong. Do not be drawn in by this. So yeah, I guess there is a fine line, obviously, as well, because some people might think that you're contributing towards those who, as you say, are not specialists in the area and who are simply accessing the online world that is not filtered in any way. And then they're coming across articles written by people from different backgrounds and with different goals and different motivations and with different messages, core messages, that it can be difficult for someone to filter through all that as well.
Mm-hmm. Yeah, I think it's also different. I mean, there's one situation in which it's an honest mistake, and I do sometimes write to people, so I scroll down to the contact me part and write to them that there is a mistake and that they should double check what they've written about. And I've had that happen to me as well when I accidentally have typos or factual errors in my articles, and I really am thankful that scientists write to me and correct me, because it is important. But if it is an ongoing miscommunication of science, then I would be more likely to call out people like XYZ. Don't listen to them. They're pushing a false agenda.
Yes. And because you're working in the field of microbiology, and obviously there's a lot of controversy in relation to the vaccines. And do you meet many anti-vaxxers in your daily life, or even in your, maybe without naming people, across your network, whether it's your private or personal network?
I think I have sort of ensconced myself in a community of scientists here in Utrecht. So in terms of like close connections, not so much. I'm not really coming face to face with people who are suspicious about it for the most part, but sure. I mean, via I do hear about, there's always the uncle or whatever of somebody else who is suspicious, right?
Yes, of course.
Yeah, and it's a tough topic because, I mean, you can never communicate effective science in one sitting to somebody who has soaked up a lot of pseudoscience or misinformation. So, Yeah, I find this a really challenging time in terms of science communication, because there's so much anti-science communication out there as well.
Well, I mean, there is. I mean, I don't know if it's anti-science communication. I think it's more along the lines of the people have got a misinterpretation of the scientific process because they've been engaging with articles that include misinformation, and they now have formed inside their own minds an idea of how the scientific process takes place and what is done first and what happens next and what happens next. And of course, there are lots of people who question the vaccine production process, who question how we can assess and quantify threats from variants. And that has been formed and it's ingrained in many people. And to reverse that, you have to, in any field of science, you have to present, first of all, the 101 elements, and then you go to, well, this is why what you've seen previously doesn't align with what we know is the case from scientific investigations that have been done over decades and hundreds of years that show us that this is the way things are. And it's very hard to fight. I think fight's the wrong word, and I don't want to call correct, but what you want to do is you don't want to convince people either. You want to present them with the information in an unbiased manner so that they can take that information, and it's voluntary, of course, with that information, whether they assimilate it or not, and then from that moment, then make their decision. You can't convince people or force people, and I think that with the misinformation that goes out there, there's a little bit of the forcing issue going on with that, I think.
Yeah, I do see, I see online there are people who are really presenting misinformation as truth, right? And that's kind of what I meant by anti-science communication. Like, these are people who are really, really suspicious of scientists who have developed the vaccine and think that they're, up to something nefarious in their work. And I mean, I do get, it is good to be critical and suspicious of what you hear, because that's also how scientists think, right? I think these are also people who, given the correct, who given more training would be excellent scientists because they're critical and they're looking for facts on wherever they can find them. And yeah, I think you've said it in a nice way that you have to give the best information from the best sources, the most reliable sources possible, and just try to allow people the space to come to reasonable conclusions themselves. Yeah,
I think that's the only way that it will work, because if you try and really force things, then you're going to alienate people, and people will feel like that you have a hidden agenda, and then you'll create even maybe a bigger gap or distance between you and them. Before we move on to the last section, the surprise section, what was the biggest lesson that you learned from your FameLab experience, and how will it influence your current research, the stuff you're doing right now?
I think it was maybe twofold. One was that this is a harder question. There are a lot of lessons. Okay, the biggest lesson. I'll pick one.
That's the question. The biggest lesson.
Yeah. I think the biggest lesson was in condensing a story into its most efficient form in 3 minutes, and strategies and how to do that. I came at this as a writer. And I came at this as a writer of a blog where I have full control over how long the articles are. So some of them are quite long. So it was a really good exercise in condensing my stories and making them snappy and interesting and only 3 minutes long. Yeah.
Yeah, that's very difficult. Doing the pitch element is very, very difficult to do. I can say I have never done a 3-minute pitch. I've recorded videos for my YouTube channel that are maybe have that kind of time constraint. I have a 5-minute video about osteogenesis imperfecta, and I put a 5-minute constraint on myself to do that. But even then, that is really difficult to make sure you tick all the boxes, and you make sure that all the various stakeholders and key points are met, and that you share all the information, that so somebody has not a diluted version of the story, but they have, as you said, the key components, the cogs of that principle or concept you're trying to communicate, and they're not distracted by the bells and whistles, that they get the absolute foundations of it. And then, thinking about that then, because you're talking about how do you create a narrative that can effectively communicate the nuts and bolts of piece for science communication. How is this going to affect your research now? Do you look at your research a little bit differently and how you approach it?
I think I always approached it from a bit of a science communication standpoint because I mean, my favorite part of being a scientist has always been talking about the science that I do. So I'm always trying to think of interesting ways in which I can construct an article at the end of all of this. So I think it's not really changed the way that I approach my research, but it did renew my excitement about my research. I think it's kind of giving me the push I need to get through the last year of my PhD. Because, yeah, it was really nice to hear that people found my research exciting, and that's, all the more reason to finish it.
Well, that's also true, but what I'll tell you now is that I know that you're entering your fourth year, and things are going to be busy. And one thing I will tell you is this, and take it from someone who's done the PhD track and knows what it's all like: don't wish the time away. Don't wish it away, because you will regret wishing it away afterwards, because you'll be like, oh, those days when I was doing my PhD, the things we used to do and the fun and the lessons being learned. And I know it's so tempting to just go, oh, I just want this finished. Try and avoid those words. I wish this was done.
I will take that with me. I have days when that's all that I'm saying, and other days when I think I have the best job in the world. So it's full of ups and downs, but it is a special time. Yeah,
lean more on that and enjoy it. And I know it's a process. You'll be like, oh, this thing didn't work and that thing didn't work, and it'll be frustrating. But do avoid wishing it away, because as you wish it away, you'll be always looking to the future, and you might be getting slightly distracted about what you're working on in the present. So live in the moment and enjoy the process. We're on to the last section, and this is the surprise section. For you, I think anyone else is going to be in this series thereafter will already know what this is. I'm calling this the Surprise Superhero Scientist Quiz. And since we're on the Superhero Scientist, this YouTube channel, there will be some superhero trivia included in this quiz. So this is how it's going to work. I'll be asking you 10 questions. You'll have 90 seconds to answer the questions. It's up to you to get as many questions right in that time before the 90 seconds is up. You can answer the questions as many times as you like. If you don't know the answer to a question, you can skip it and you can come back to it later. There'll be some science questions related to your field. There's even a question outside of your field, because I need to make it a bit more diverse. And there are some superhero questions in here as well. Whatever happens, because this is number one, you're going to be top of the leaderboard after this, as you're the first person to take part. So the aim here is to try and get all 10 questions right in the shortest time possible, and then we'll be able to look at what, if you do it in the 90 seconds, for example, and you get 8 right, then that's going to be your answer. 8 questions in 90 seconds. Do you understand?
Sounds good. I love quizzes. I'm in.
Good. So what I got to do here is I got to get my stopwatch ready. So for all of this, so that I can make sure I'm keeping an eye on things. Going to bring the questions in here beside me. I was going to put pictures in this as well, but I think it'd be easier for me to just ask them. I think you're going to get some of these questions straight away, but you have 90 seconds. If you don't know a question, I will be putting it down, and then we can come back to it. And I'm going to start everything in a moment. Are you ready, Alex?
I'm ready.
You're ready. Excellent. Right. Okay. 1 second now. And my phone here has decided to unlock itself, or to lock itself. So let's make sure that doesn't happen. Right. Are we ready to go? We are?
Born ready. Yeah. Okay.
Yeah, born ready. Come on. Oh, that's a thick speech. I said cringe earlier on. That's such a cringe statement to say. Right, so 3, 2, 1, and we're off. Question number 1. Name the 4 nucleotides that make up DNA.
ACTG. Shit. Adenine, cysteine, threonine, guanine.
Great, that's that one correct. Question number 2. In the Avengers film from 2012, which superhero does the actor Jeremy Renner portray?
Oh, I'm terrible with actors' names. Skip.
Skip.
Okay.
We are currently dealing with the Omicron variant for SARS-CoV-2. Omicron is the 15th letter in the Greek alphabet. What is the 16th letter in the Greek alphabet?
Skip.
In number 4, including Spider-Man No Way Home, which releases in December 2021. How many films has Tom Holland portrayed Spider-Man in?
3?
No, you
4.
can give another answer. No, you can keep on thinking about that. No. Question 5: Which superhero film made more money at the box office? Avengers: Age of Ultron or Black Panther?
Black Panther.
That's a wrong, so I can't give you that as an alternative. Yeah. Number 6, you're going to get this. What does PCR stand for?
Polymerase chain reaction.
Correct. Besides Crick and Watson, who also helped to decipher DNA but died in 1958 and never got full credit for the work?
Rosamund... forget her last name.
I'll give you Rosalind Franklin.
Rosalind.
Which species...
Yeah.
Rosalind Franklin, yes. Number 8, which species has a larger genome in base pairs? A human or a woolly mammoth?
Woolly mammoth.
Yes. In physics, what is the unit for magnetic inductance?
Ohms? No, that's resistance.
You can keep
Skip,
guessing on that. It's 10. Yeah, skip. Question 10 and the last question. Which character speaks first in the film Avengers Endgame from 2019? Here's a clue. I mentioned the character earlier on in the questions.
Spider-Man.
No. Okay. I'm gonna stop it there because actually it's good to know this because this was the first go. You need more than 90 seconds. So that was 2 minutes. I'm going to say that was 2 minutes. Let's take a look at what you got and what you didn't get. You had the 4 nucleotides. That was great. That was fine. Number 2 in the Avengers film from 2012, which superhero does the actor Jeremy Renner portray? That's Hawkeye. Number 3, you're going to kick yourself with this one. Omicron is the 15th letter in the Greek alphabet. The 16th letter is pi.
Oh, man. Yeah.
Yeah, MNOPOPQ.
Yeah.
Number 4, the number of films, including Spider-Man No Way Home, which is out soon, or in December 2021, has Tom Holland. That's 6 films he's been in. You had 4, 3,
of course,
and 4.
because the Avengers film. Yeah, okay.
You could have kept guessing there. Number 5, the superhero film which made the most money at the box office was Age of Ultron. It made 1.4 billion. Black Panther was 1.35. Number 6, you got PCR. Number 7, you got that as well, Rosalind Franklin. I gave you that because you knew the first name, and a lot of people don't know who that is. Number 8, which species has the largest genome? Correct, you said the woolly mammoth. Number 9, in physics, magnetic inductance, the unit is the Henry. That's a hard one,
nice.
But I had to give you a question outside of your area. And number 10, the last question, which related back to all the other questions, which character speaks first in the film Avengers Endgame from 2019? And that was actually the answers from answer number 2, which is Jeremy Renner played Hawkeye, and he was the first character to speak in that film. So,
tough
going
quiz,
back, tough quiz, but unsurprisingly, of all the questions in your own area, you got 4 out of 5 on your own field. So,
that's a pass. Yeah,
that's a pass. That's a pass. Yeah, not in the Netherlands though. It's a 5.5 in the Netherlands apparently, or a 5 or something like that. But we'll see in Ireland and I think in Canadian educational systems, that's a pass. That's a pass. Yeah. Let's go with that. That is a tough question. Also good for me to know that I need to give people 2 minutes and not 90 seconds because you need that little bit more extra time to think. Right, have you learned some valuable lessons about science and superheroes from that quiz?
Definitely, that I need to rewatch the Avengers films, and I'm going to look into why the unit is called a Henry later today.
Yeah, well, it's obviously it's named after a guy whose surname is Henry, so that's the big
spoiler.
The big giveaway
yeah.
there. Big spoiler on that. Before we finish, just so people know about you and what you do, you have your blog, Microbial Mondays, right?
Yeah.
Where can people read your blog?
Microbialmondays.com. Easy to remember.
Very simple. Super simple. And you try and write every Monday when time permits, right?
Yeah, pretty much every Monday now. I'm trying to be a little more consistent on it. And there will be a link to my up-and-coming YouTube channel there as well. Right.
Good. Excellent. Yeah. What's the channel title going to be? Is it going to be the same Microbial Mondays as well or something else?
It's Alex Goes Viral, and Microbial Mondays will be a series on it. So maybe more series coming in the future.
Okay. Very, very good. Anything else anyone needs to know about your research? If they want to know about your research, where can they get information on that?
Good one. Also on my blog, there's an about page which has more information about my research and links you can follow to find my manuscripts if you really want to dive into the data.
Fantastic. Excellent stuff. Well, Alex, thank you very much for the super interesting discussion in relation to your work, in relation to your experience with science communication, in relation to your FameLab experience and your success in 2021 with FameLab, and also getting the chance to have you take part in this first quiz on science crossed over with superheroes. And you've set the mark now of 4 questions in 120 seconds. Let's see how
of a fail.
Other people get on with it. It's not a fail. It's a pass. At the moment, you're top of the leaderboard.
Perfect.
So there you go. And I'll be putting a leaderboard in here. So here it is. There's the leaderboard. There you are, right at the top with your 4 correct answers in a time of 120 seconds. There you are. So you can say you're number 1 for the moment until somebody else does the quiz. Alex, thanks for the time. Really appreciate it. And best of luck with everything you're doing.
Thanks so much for the chat, Barry. Always a pleasure to talk about science and superheroes with you.